And I'll call on our first speaker. While I talk a little bit about him, maybe he'll connect his computer at the same time. Professor Dani Peer is a professor and the director of the Laboratory of Precision Medicine here at Tel Aviv University. This one. He's also the director of the Leno and Harry Helmsley Nanotechnology Research Fund and a chair at the Tel Aviv University Cancer Biology Research Center that includes 17 affiliated hospitals. Professor Peer's work was among the first to demonstrate systemic delivery of RNAi molecules using targeted So it's his work is pioneered in the immune system and uses RNAi, which is RNA interference molecules, for in vivo validation of new drugs targeted within the immune system. He has over 50 patents, and some of them also developed into spin-off companies. It says here 4 spin-off companies. And he generates, he aims to bring personalized nanomedicine into clinical practice. Thank you.
Okay, so I'd like to thank the organizers, mostly Mira, for giving us the opportunity to talk a little bit about our work. And I'm going to start with really basically trying to explain to you, what are we doing, but before that, how do we really think? And in order to do this, I have to start with an example. And the thing is that you have to really think a little bit outside the box. And how do we, as a scientist working in this very multidisciplinary area, how we really can try to solve problems? And in order to do this, I'll start with an example. Let's assume a car is fallen into the river. And now you need to take it out from the river. And there is many options, many possibilities how to do this. But the majority of people think that if a car is falling into the river, what you need to do is to grab it out, bring a car. So this is the car that is fallen into the river, and there is a bigger car that is now trying to get it out from the river. But only very sharp people, or physicists, will actually see now that this car is full of water. And therefore, all the momentum here is miscalculated. And what will happen is most likely this. And now instead of one problem in the water, we have 2 problems, right? And what is the classical solution for this? Bring a bigger car. And what will most likely happen? This. Okay? So this is the circle. We are living in this very interesting circle, and we have to break it. We have to think outside of the box. And I would like to give you some examples, or maybe one example, because we are short in time, how to do this. I'm going to talk a little bit about our lab. So our lab is investigating how to manipulate cellular function in order to generate novel therapeutic approaches to treat mainly immune system-related diseases, which is a very big field that includes blood cancers, which is here, inflammation, and viral infections. But it also has meaning to other solid tumors, ovarian, brain tumors, rare genetic diseases that relate to the immune system, and melanoma, but we are not talking about melanoma. This is why we have Karmit here. So I'm going to focus only on blood cancer, and the lab, in our case, is divided into 2 major groups. People that are doing basically discovery concepts using genome sequencing, exome sequencing, DNA sequencing, and RNA sequencing to identify new therapeutic targets. And then people that can deliver those new therapeutic targets specifically into specific cell types. And again, we are looking at leukocytes because immune cells are really challenging. The idea, of course, is to combine those new discoveries from this arm and this arm into one carrier that could be one drug. And we personalize this as the way I'm going to show you now. But before I'm going to give you one example, and this will be an example in blood cancer, I want to say that we are moving into the precision area of medicine, basically the ability to sequence the genome, look at the mutations, basically also look at the RNA level, provide an opportunity in a very dynamic disease such as cancer to follow up and be in this war, in this battle, very, very close every time to offer new treatments. And the idea is to sequence patients, learn about their mutations and genes that are either overexpressed or downregulates, and design and synthesize new drugs, most likely based on RNA, that could be either messenger RNA, if you want to upregulate a gene, or siRNAs, if you want to knock down a very specific gene or a gene area that fits patient profile. Unfortunately, this part is the easiest part. It's not easy, but it's a doable part. But then you have to package those molecules that are super negatively charged and deliver them only to the disease cells. And for this, you need vehicles, kind of a taxi or Uber, that will take you to the appropriate area. And we're trying to do all of them. And my example will be one single example, but for us, again, it's a proof of concept because we are involved in different kind of diseases. The example I want to give you relates to mantle cell lymphoma. It's kind of a rare disease. About less than 6% or close to 6% of the non-Hodgkin's lymphoma. It's considered an incurable disease with traditional type of chemotherapy. And basically, it's a bone marrow kind of disease. From the family of hematological B-cell malignancies, with the poorest among B-cells lymphoma. And you can see here a PET CT of a patient that have a lot of black dots, which are metastases. And again, this is a bone marrow disease. You can see this patient is in a very bad shape. Today, there are new drugs approved for this disease, which extend the median survival dramatically from 3 years to about 5 to 7 years. Still, it's among the poorest in the B-cell malignancies, or the B-cell lymphomas. But this disease has a very unique genetic profile. In about close to 85% of the patients, there is a chromosomal translocation. The gene that encodes for cyclin D1 juxtaposed into an area in the genome that is now starting to be produced. And this gene that is called CCND1 that encodes a protein named cyclin D1, which is responsible for proliferation of both normal and cancerous cells, is now being expressed, dramatically expressed. Normal B cells usually do not express a lot of, or at all, of cyclin D1. There are other type of D-type cyclins. So potentially, this is a kind of a rare disease. In Israel, we have about 12 patients at any given time. So really, really rare. In the States, about 3,000 patients overall in any given time. So you have a unique opportunity to actually destroy this gene. And we have sequenced 80 patients, both from Israel, US, and Europe, and basically revealed that there are 2 transcripts in their cells. It's also inaccessible. You can actually take blood samples and isolate those B cells that are sick, diseased B cells, lymphocytes, and really look at the transcript. And we have found that the patient have 2 types of a transcript, of messenger RNA. One is a short form and one is a long form. That means that if you want to now develop a drug to silence, to make sure that these genes is not produced, or basically the protein is not produced, you need to design it in an area that will target both type of messenger, the short one and the long one. And a few years ago, we started a collaboration with a Boston company named Illion Pharmaceuticals. And together with them, we screened different sequences of siRNAs against this gene and have shown together with them that we can knock down very effectively in cell lines and in patient samples and basically create a cell cycle arrest that will eventually cause a cell death. So potentially now we screen, it took about 3 years, we screen for a potential drug. Is it going to fit all patients in all fields? No. It's going to be narrowed down only to the mantle cell population. However, in other malignancies, B-cell malignancies such as multiple myeloma, 25% of them has the same translocation. And in other type of cells, like CLL, chronic lymphocytic leukemia, some patients do have this translocation. Might this fit to them? Maybe. But now we had another challenge. Okay, so you have a potential drug in hands. How are you going to administrate this to the patient? And how are you going to make sure that it's going only for the diseased cells? So there is a lot of challenges in this field. And you see, the problem is that leukocytes in general are very hard to transduce or transfect with nucleic acid. They're spread around the body, and there are very specific requirements. In general, normal lymphocytes, it's the majority inside is the nucleus. And we know that nucleic acid, like siRNA, are working only in the cytoplasm. And if it's activated, it's about 75% is the nucleus, and you don't have a lot of cytoplasm. So you need an efficient encapsulation of those nucleic acid. You need evade clearance mechanism. You need to avoid toxicity and immune activation. You need to create specificity when you inject this into the bloodstream. And you need to have a carrier internalization. So if we encapsulate those drugs, those nucleic acids, short interfering RNA, into a carrier, this should be a small carrier and highly selective. You need actually a GPS on this because you have to direct it into the cells that needs this systemically. And then, of course, when this carrier enters the appropriate cell type, it needs to effectively and very fast release its payloads. So you have multiple challenges on the way. So how do we start? So these are basically the work of two talented graduate students, Dr. Shiri Weinstein, who is also almost a physician, did a PhD, and Itai Toker, who is a PhD student. And their aim was to develop this highly selective particle that can target mantle cell lymphoma cells and deliver those therapeutic siRNA against cyclin D1. And they built it on a strategy that we published a few years ago, but they improve it. First, we had to create a model, a mouse model that will recapitulate the disease. And we were amazed from the fact that there were no mouse model for mantle cell lymphoma. Usually people are taking the cells and inject them subcutaneously. So they stably transfected cells with GFP and have shown that if they inject the cells systemically, the majority are accumulating in the bone marrow. And this is just an example of a normal bone of a mouse and a mouse that were injected systemically into the tail vein of those cells. And you can see that the tumor is forming inside the bone. And this is very similar to the human disease. In generally, this is a very fast process, and in less than 30 days, those mice will die. The question is it a good model? It's a very aggressive model. Then we have to define a targeting moiety, basically a GPS system. Can we put something on the surface that will direct those tiny nanoparticles made from unique lipids directly into the appropriate cells? And we screened for different receptors that are fast internalizing receptors. One of them is CD22, one of them is LFA1, and the other one is CD38. And we found that in at least cell lines, this is the reaction. So basically, we wanted to use this one, an antibody against CD38 as our targeting moiety. Then they took the antibody, labeled it, and basically showed both systemically that when you inject the antibody, it goes to the bone marrow. And also, if you sort out the cells, you can actually see the antibody is internalizing. And that was the first evidence. Exactly at the same time we published this paper about a year ago, FDA approved this antibody for multiple myeloma as a new therapy, just the antibody, because it has a very unique biological activity. But we used it as a targeting moiety. So it happens basically at the same time, but this is the first manuscript that actually we published that showed the mechanism behind this. Okay, so we generated very unique lipids, which we call ionizable lipids. And using microfluidics, we actually streamline a process development where we can put nucleic acid, in this case siRNA, lipids in ethanol, and basically mix them on a mixer and get very fast, tiny particles, and then we can decorate them. The idea of those lipids are that we can, these are ionizable lipids, that they are charged. When we put mildly acidic environment, they are now positively charged, and you know that nucleic acid are negatively charged, so that will be a great condensation on this mixer. And then at the end, we change the pH to neutral in charge, and now their total charge is neutral. So we don't get immune responses, and we avoid many other types of toxicity. And this strategy also streamlined the process. We can actually scale it up to a liter capacity, and this is already therapeutic. So basically, you can make sure that if you do it under GMP condition, on a liter scale, you can inject them to patient. And we developed this strategy. We also published it. But basically, the idea was we used the microfluidics as a way to streamline and create mass production in a very robust manner. Then what about the targeting moiety? So for this, we had to use other strategies, and we have to purify. We put the antibody on the surface, we decorate the surface covalently, and then we have to purify this. So this is not an industrial process yet, but we are working on this part. And you can validate then the specificity, the efficacy. You get tiny particles, about 100 nanometer in diameter, and you can actually inject them. And this is just an example of the specificity from 2 different patients and from a co-culture system with the cells that have the receptor, cells that do not have the receptor, and it binds, internalize, and silence. And this is just, again, a fluorescent using flow condition, basically that non-targeted particles do not even internalize, particles that have an isotype control not internalize, and basically you can deliver your payload with a specific antibody. Again, under shear flow condition. And we have shown that we can silence the gene and also block the cell cycle in the appropriate area. But the big question is how specific it is and can it use for therapeutics. So we did an experiment, a blinded experiment with a company that didn't know exactly what they're getting, and these are the results. We injected, and you can see extend survival dramatically with this. But you can also see that at the end, all the mice are dying. Okay? So this is a very aggressive model. And what we found on this is that it's not enough to block only one pathway, which is very logical. We have to start doing combination therapy, and this is exactly what is happening in the clinic, right? The patient gets a combination of drugs, not a pathway-specific only drug. And this is the area that we are now doing, or the area we are looking at, is combining different types of pathway in a single nanocarrier, because it seems that the nanocarrier is very specific. And now we can take, again, the samples from the patient and see exactly where are we going and how efficient we are going to be. At the end, there's a platform. And as a platform, you can put any antibody as well on the surface. And you can encapsulate any types of nucleic acid. Majority of them will be, it depends on their size, but basically it's a doable platform. And the people that are really working on this, I think these are the most important. But my take-home message, it's all personal and it's all in the message. So if we can quantify the message and the gene, and we can play around with this, we really will be able to create personalized approach, which is already something that is starting to happen even in the clinic. And just in Germany, a few months ago, 50 patients were injected based on their own discoveries of neoantigens as a kind of a vaccination approach. But it's a personalized and a very specific approach. At the end of the day, I want to thank the funding agencies and the people who really work on this in general, and specifically the people who did the work that I already mentioned. Thank you so much. Questions?
Yeah, we have a few minutes for quick questions. Anybody has?
Yes.
You're talking about a nanoparticle that finds its way. It has some sort of internal GPS or router. What's the efficiency rate? I mean, how many units do you have to inject to get into one cell?
So the question was, how many particles we need to inject in order to get into one cells, right? One cell. And the answer is, we really don't know. We know in vitro on cell culture how much we need to put, concentration ways. In the next level, we know in mouse how much we need because we can measure it. In humans, this is our next step, we'll need to measure it and titrate it. And the phase 1 studies are usually designed to create MTDs, a maximal tolerated dose, and then the efficacy, it's only next in line in the next phases. So this is a process that you need to learn. We don't know yet. We know how much time it takes for the gene to come up because it's a transient silencing. It's not forever. So we know that if the cells are dying, the new population that's coming back could take a few weeks. So we imagine that the injections like this will be once a month in general, or even once every 2 months.
Okay.
Okay.
Thank you.
Thank you.